Hi, this is Eric Hayden from Boise State University, and today I'm going to tell you about some research from my lab that focused on the evolution of new RNA folds and functions. So if we imagine an RNA world where RNA played an important role in the origins of life, then we can imagine the need to evolve from a simple replication system on the left to something like a last universal common ancestor on the right. And this process must have involved numerous evolutionary innovations, which are the emergence of entirely new folds and functions through the process of evolution. And we have some good experimental systems to study the optimization of a given function in the lab, but we don't have a lot of experimental systems to understand the evolution of entirely new structures. One concept that's useful to think about when thinking about innovations is the idea of a genotype network. And so a genotype network is the collection of all the genotypes that can form the same structure and that are connected by individual mutation steps. So in this hexagon on the left is a representation of some computational results where all of this, the hexagon is genotype space, and the black nodes indicate genotypes that all form this hairpin on the left. And you can see that it's possible to walk from the left side of the space to the right side of the space, completely changing the sequence without ever changing the structure. So these genotype networks can be very large and very connected. And this is also backed up by what we see in sequence conservation in nature, such as this 5S ribosomal RNA sequence conservation map on the right. And so the nucleotides in red are greater than 90% conserved. And the nucleotides shown as white open circles are less than 80% conserved. And so you can see that there's, since the ribosome only emerged once through evolution, it's diversified incredibly over time. And while the structure and function has been maintained, the sequence is completely diverged throughout the bacterial domain of life. And so in terms of innovations, we can imagine that innovations can happen where two genotype networks come in close proximity or even intersect. So we have a blue network here, which represents one RNA structure on the bottom of the slide, and a purple network that represents a different structure. And so we can imagine starting at the node 1, which is one sequence, and we can take a mutational step to 2 and another to 3 and so on. And we might end up in a point on this network where we end up in close proximity to the other structure, and a single mutation or very few mutations can change from one structure to the other. And so we wanted to study this yellow region in the lab where two genotype networks come in close proximity. And we had a good experimental system to start from. So about 2 decades ago in the lab of David Bartel, Eric Schultz was sitting around basically playing Eterna by himself on paper. And so what he noticed was that there was this ligase ribozyme and this HDV ribozyme that they were the same length. And so he imagined he could find a sequence that would satisfy all the base pairs for both structures. And that's in fact what he did. And he found this single sequence, which is called the intersection sequence. And it is capable of forming the base pairs of both structures. And when he took it to the lab and made it, he found that it indeed had low but detectable activity for both ligation and self-cleavage activity of the HDV ribozyme. And then he further made intentional mutations that favored the HDV fold in red and showed that he could connect all the way to the prototype HDV sequence, or the wild type, through a series of mutational steps. And likewise, he could proceed from the intersection towards the ligase prototype and maintain function the whole way. So this clearly demonstrated that these two networks, there's at least one trajectory that leads from one genotype network to the other. But we wanted to zoom in and find out about all the other numerous trajectories that aren't present in this data. So we chose a reference ligase sequence and a reference HDV sequence that each had high relative activity for their given function, and were only 14 mutations apart. That enabled us to make all the combinations of these 14 mutations, which required the synthesis of about 16,000 different RNA sequences, and enabled us to study not one trajectory, but 14 factorial trajectories, which is about 87 billion trajectories between these two reference genotypes. And importantly, we wanted to know, just like the Bartel lab did, of what is the mutational consequence of each mutational step on these trajectories. And so we set out and we designed and synthesized this combinatorial library, which contained combination of 2 nucleotides at each of 14 positions, shown as the red and blue letters. And we've shown these mutations on the HDV fold on the left and the ligase fold on the right. And we used a high throughput sequencing assay to detect the fraction cleaved in the HDV fold and the fraction ligated in the ligase fold for all 16,000 of these sequences. And so our data looked something like this. And what we found is that over here on the left side of the network graph in panel A is the HDV structure, and the sequences that have high HDV activity are in red. And on the right side of the network graph is the ligase reference sequence, and sequences with high ligase activity are in blue. And both the size and color intensity of these nodes represent how active these ribozymes were. And in both this bird's eye view on the top in panel A and from the side view in panel B, what you can see is that as we move away from the HDV sequence and acquire more mutations, we eventually get to a point where this activity kind of falls off. And likewise, if we start from the ligase sequence and move towards the HDV, the ligase activity falls off. And right in between, there's sort of this valley of death, but it appears there might be numerous intersection sequences. And in fact, when we looked for all the sequences that could catalyze both reactions, we actually found over 9,000 intersection sequences. And I would say this was a little surprising. And what was also surprising is that while many of the sequences have very low activity for both functions, some of these sequences had relatively high activity for one function and still had very detectable activity for the other function. And so those are the sequences sort of shown up in the blue quadrant, had high ligase activity, and those in the sort of pink quadrant had high HDV activity. And so these seemed like likely candidates that would support the evolution of new functions. So to understand this evolution better, we developed computational simulations of populations evolving on these genotype networks. And so I'm not going to get into it too deeply, but you can go back and look at the slide later and see the details in our publication. But essentially, this model allowed for numerous individuals to be competing for survival on these landscapes. The probability of survival was stochastic, but was based on the fitness of each genotype. And mutations happen also stochastically. And so the population tends to move towards higher fitness over time. So we imagine this scenario where we first had an HDV ribozyme in sort of a proto-organism, and we had a gene duplication event, and then the second copy of the gene was free to evolve towards ligase activity. So we started from genotypes in red that had high HDV activity, and then we challenged them to evolve on the ligase landscape. And we recorded these populations evolving through this evolutionary challenge. And the recordings look something like this. So the size of this blue circle indicates how many individuals are at that genotype. And so this large blue node to start means that they all started at the HDV reference, and we're going to let them evolve towards this ligase peak activity. And the generation time is on the top. So as we evolve, the population moves as competitions for survival happens. And eventually, the population finds its way to the peak of the ligase activity. And it takes about 300 generations to get up to that peak. If we restart the simulation and put all the starting individuals at a different genotype that are 2 mutations away, we see different dynamics. And we see, in this case, that evolution goes very quickly to the top, and there is no pausing at these low fitness. And so in this case, the population finds the peak in less than 100 generations. However, if we start from a slightly different genotype that's also 2 mutations away from the prototype HDV, we can see that adaptation goes very slowly, and this population gets stuck for a very long time in one spot. And in fact, in this simulation, it doesn't even ever escape from this local fitness optimum. And so we did this from numerous genotypes, all the genotypes that had HDV as active as HDV or more active, evolving towards the ligase activity. And we did the other way, taking the same number of genotypes on the ligase side and evolving them towards HDV activity. And we see these different dynamics depending on where we start in genotype space. And these are all individual simulations, but we also see this when we average hundreds of simulations together. So that's what's shown on this graph on the left, where the blue traces are the average dynamics of about 100 simulations, and each blue trace starts from the same genotype. So we can see that those blue genotypes tend to have very rapid adaptation, whereas the red genotypes, if we start from those genotypes, they tend to have very slow adaptation rates. And when we looked at where these red traces, where they're getting stuck and where they're pausing, we find that they get stuck at these local fitness peaks shown in green. And so these are peaks where they're genotypes where all of the distance 1 neighbors have lower fitness than themselves, and even most of the genotypes 2 mutations away also have lower fitness. So evolution tends to hit these peaks and get stuck there for a long time. And so hopefully I've demonstrated to you sort of a very first quantitative glimpse into evolutionary innovations. We found that intersection sequences may be much more common than previously appreciated and may actually serve a very important role in the evolution of new RNA folds and functions. Hopefully we've shown you that these evolutionary simulations are a useful way to analyze these fitness landscapes. And I think another message is that fitness peaks, not valleys, are the key control for adaptation rates based on these simulations. With that, I'd like to thank my funding sources and thank all of you for your attention.